Hola.
Hello.
Hello. Hi, everyone. Hope you're safe.
Hello. Welcome.
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 Welcome. Welcome.
Hi, PRISM students. We miss you, and we're very excited to hear about your research projects.
Hi, how are you? This is Dr. Chen. I miss you. During this busy time, you're doing a fantastic job. I'm so proud of you.
Hi, everyone. We miss you a lot.
Hey, everyone. We miss you.
Don't be nervous. Good luck on your presentations.
Wishing you and your family peace and tranquility during these difficult times.
So I'd like to share with you a quote that's helped inspire me on those days when I'm feeling down. It's from the Winnie the Pooh series, and it's something that Christopher Robin tells Winnie. Christopher says, "You're braver than you believe, you're stronger than you seem, and you're smarter than you think."
Hey.
Hi, everyone. Before we get started, the PRISM team and I wanted to tell you how proud we are of all the work that you have done to help us put together this symposium. And to tell you that we miss you and that we cannot wait to see you again when things get back to normal. And although we cannot be in the same place right now together at John Jay's Dining Hall, sharing a delicious meal provided by MBJ, we want to remind you that we're all in this
Together.
And now, allow me to introduce John Jay's Dean of Research and the Director of the Program for Research Initiatives in Science and Math, Dr. Anthony Carter.
Thank you, guys. That was awesome. I've seen that video many times now, and it always chokes me up a little bit. Welcome. Let me echo what was just said. We miss you all. We miss having you guys stopping into our offices, working in the labs, and just generally brightening our days. We really hope you all are well and safe. To say it's been a challenging semester is possibly the understatement of the year. I'm so proud of everyone participating today. Proud of our faculty who have not only moved their classes online, but have figured out ways to meet with and mentor their students from their homes. Proud of our students for continuing to charge forward with their research and putting together wonderful electronic posters that we'll see later in this session. And proud of our staff who have literally moved mountains to put our services and this symposium online. Let me actually say a special thanks to our staff, including Gabriela Sanguinetti, Derek Sokolowski, Erica Clayfin, Leslie Porter-Cabell, Pat Samperi, Oscar Fuentes, and of course, the man who pulled this all together, Dr. Edgardo Sanabria Valentin. Our staff have been working round the clock to help students create slides, write scripts, and practice for this event. So I'm hopeful that in about 3 hours or so, when we're all done, Ed's blood pressure will start to return to normal or so. I want to acknowledge some volunteers that have helped today. Rachel Friedman from the Office for the Advancement of Research and Jonathan Lee Gibson from the Law and Police Science Department. Let me give a big shout out to the college for helping us move this event online, especially the Office for the Advancement of Research, the Office of Marketing, the Department of Information Technology, and the President and Provost's Office for supporting this event. Let me thank the Association of Program Administrators of C-STEP and STEP and its executive board, who provided some of the technology to run this event. And finally, let me give a special thank you to the U.S. Department of Education Title V program and the New York State Education Department's C-STEP program, who have made PRISM possible. By last estimate, we were expecting several hundred people to join today. Students, faculty, families, colleagues, other special guests. Welcome all. Let me give a special welcome to Brooke Gesser from Senator Kirsten Gillibrand, who is a great supporter of the public institutions higher learning in our state, CUNY and SUNY, and Kim Overrocker and Joanna Heinsohn from the New York State Education Department Office of Post-Secondary Access, Support, and Success, who are always ready to provide us the services that allow us to prepare to succeed at John Jay and prepare for their professional lives. This has been a difficult time for all of us. Everyone has been affected in some way. We have friends or family who have had the virus. We have loved ones who are working essential and high-risk jobs, some who have lost jobs, and others who have lost loved ones. We've lost one of our PRISM family members to COVID this semester, and we'd like to dedicate it to Dr. Donald Hoffman. Don was a long adjunct of sciences who worked with us for more than 40 years. Don helped establish the toxicology track within the major. He taught many generations of toxicology at the college, and he loved to work with students. For many years, Don served as a mentor to PRISM students and judged our poster award competition. Those of you who knew Don also knew that he always mingled with students during the dinner portion of the symposium. As a matter of fact, Ed was sharing an anecdote with me just the other day that feels very relevant. When Ed took over the organization of the symposium in 2015, he had not yet met Don and was struggling to line up faculty who could serve as judges that year. 2 weeks before him, Don walked straight into the PRISM office, went up to Ed and said, "I hear you're the new PRISM guy. I've been a judge in the symposium since it began, so where do I sign up?" Since then, Don was the first person that contacted Ed to serve, and he never failed to be there for our students. Don will be missed, so please join me in a moment of silence to remember our colleague, teacher, and friend. Thank you all for your resilience in coming here today and for presenting your work. It really is an incredible testament to our students and faculty that we're able to put this online. Now let me introduce our next segment. Every year we ask faculty to nominate students from within their labs for our Outstanding Undergraduate Researcher competition. These nominations are reviewed by a small group of PRISM staff and faculty, and they have the difficult task of selecting the best of the best. The Outstanding Undergraduate Researcher Award recognizes a student who's excelled in the lab through their commitment to research, but it also recognizes a student who has shown exceptional personal and professional growth during their time in PRISM. This year, I'm delighted to introduce John Jay faculty member, Dr. Marta Concheiro-Guisan, who will introduce her student and our Outstanding Undergraduate Researcher, Alejandro Ocampo. Alejandro will answer questions after his talk. If you have questions, please use the Q&A button below your screen to write them and send them in, and our host, Ed, will pose these questions to Alejandro at the end of our session. Mark?
Okay, thank you so much, Dr. Carpi. I'm honored to introduce my student, Alejandro Ocampo, as the 2020 Outstanding Undergraduate Research Awardee. I have been Alejandro's mentor for more than 1 year in the toxicology laboratory, and it's been a real pleasure working with him. I decided to nominate him for this award due to his excellent qualities. Alejandro is extremely mature, intellectually brilliant, and mentally strong. He is without a doubt the kind of person that you can rely on. His research project he's presenting today was a very difficult investigation that required a combination of hard work on the bench and a deep understanding of the current literature. As it always happened in the laboratory, experiments often did not work out the way we had originally planned them, but Alejandro did not faint. Instead, he showed extraordinary resilience and optimism in the face of adversity, coming up with practical solutions to move the research forward. Besides his outstanding performance in the lab, I had to nominate him for this award due to his exceptional determination and effort. Throughout the year, Alejandro has been able to successfully achieve research and academic excellence, and he has made them compatible with a full-time job outside of John Jay. This imposed on him an extremely challenging and high-demanding schedule. There were many late-night shifts, many early mornings, and many busy weekends, but I never heard him complain about any of these. When Ed first told him about the award, his initial reaction was to turn pale and exclaim, "Me? Are you sure? It's not possible." In fact, I can imagine him listening to this brief introduction right now and maybe thinking, "Come on, Professora, you are exaggerating. I'm not the person you are talking about." Well, guess what, Alejandro? Yes, you are. Being humble is a great asset, and you should be aware of your huge potential as scientists. I'm certain that you are going to succeed in pursuing a successful career in science because you have all that it takes: maturity, serenity, enthusiasm, and strong brain power. Because all of this, it is with deep pride and great pleasure that I introduce you to the 2020 Outstanding Undergraduate Research Awardee, Alejandro Ocampo.
Thank you so much for that. I need a tissue after that one. No, those words really touched the heart. Thank you so much. They really mean a lot. And hello, everyone. Once again, my name's Alejandro Ocampo. Thank you all for being here. And to my fellow PRISM students, our day's finally here, where we get to celebrate and really display our years long of hard laboratory work. Laboratory work with a common goal of answering an array of interesting scientific questions. I really like to thank the PRISM staff and committee for having selected me to be this year's Outstanding Undergraduate Researcher. It's an award that truly has humbled me. The PRISM program has changed my life in many ways. From how I behave in the lab, it's giving me experience in the technical operation of a scientific laboratory. And already it has opened doors for life after John Jay that would not have existed if I've never joined the program. So for all that, I really am really grateful. But that experience gain could not have been possible without my mentor, Dr. Concheiro-Guisan, who has taught me so many invaluable scientific skills and techniques that not only helped me tackle my project and were useful in the other classes I was taking alongside the project, but techniques that I know I will be using in my future career. And thank you so much for all that. And without further ado, I'd like to speak to you all about my project. My project So sorry, I think I'm having a difficulty here. Doctor, do you see the?
We see the slides, Alejandro. So yes. I think you could go forward.
Thank you. So sorry. My project investigated placenta as an alternative matrix to detect cannabis exposure during pregnancy. But what is cannabis? Let me tell you a little bit about it. Cannabis is a plant. It grows naturally in nature. You may have heard to it referred to as marijuana, weed, grass. It's the most commonly used illicit drug in the United States. The most notable effect and the most common reason for why people use it is euphoria, which is commonly known as feeling high. But there are other effects too. They include increase in appetite. It produces a sedating feeling, which is a feeling of calmness or sleepiness. And it could also lower your inhibitions. So you usually don't jaywalk. If you're on cannabis, maybe you'll throw caution to the wind and you'll jaywalk. Cannabis can also cause negative mental health effects, like making one more anxious and paranoid. It can decrease your motor skills, making driving or operating heavy machinery dangerous. The chemical compound THC we see on the top right is the main psychoactive chemical component of cannabis. And psychoactive is a term that describes a drug that is capable of affecting the mind and brain function. An interesting fact about THC actually is that it exists at levels so low in the raw plant that eating raw plant won't actually cause the high. The compound THC, or tetrahydrocannabinol, comes from another compound called tetrahydrocannabinolic acid with high heat, such that as in burning or in smoking, and the heat in cooking when making foods such as brownies with the cannabis, the acid species is converted to the THC, the tetrahydrocannabinol. These two compounds are compounds known as cannabinoids. Cannabinoids, they're a class of chemical compound specific to the cannabis plant. Some naturally occurring in the plant are seen here on the left-hand side. They are THC, CBN, and CBD. You may be familiar with CBD, or its full name, cannabidiol, since it has increased in popularity in medical research in recent times. And also many nutraceutical companies have mass-produced natural products out of it as well. But for this project, we focus mainly on THC. When cannabis is consumed via inhalation, the chemical compounds enter the lungs and get absorbed by the alveoli of the lungs and enter the bloodstream. THC is able to pass the barrier we have between the body's circulatory system and the brain, called the blood-brain barrier. It then affects cells in the brain that have to do with pleasure. And through time, the THC re-enters the bloodstream and travels to the liver, where it undergoes metabolism. And metabolism of a drug is a form of transforming the drug to compounds that can be easily excreted by the body. On the right, we can see some of those transformed compounds, and we call them metabolites. We call them metabolites because they are products of metabolism. These cannabinoids are found at detectable levels in a cannabis user, which is why they have been hand-selected for this project. Now, what is the legal situation of cannabis in the USA? And we discovered from further research that as of 2019, 33 states in the US and Washington, D.C. have passed legislation legalizing the medicinal use of cannabis. And 11 of those 33 states and Washington, D.C. have gone even further and legalized the recreational use of cannabis. And with this increase in legalization across the country, there has been an increase also in cannabis consumption during pregnancy. Medical surveys have been taken throughout the years on drug use. And according to the 2017 National Survey on Drug Use and Health, 7% of pregnant women used cannabis in the past month from the time of the survey. This is an almost 4% increase from the 2003 survey. But cannabis isn't the only choice in drugs used during pregnancy. The graphic shows the reports among pregnant women of substance use in the month prior to taking a survey. If we look at the top half of this graphic, we can see that tobacco was the most abused substance among pregnant women. Next comes alcohol, followed by illicit drugs. But for this project, we'll focus on the illicit drugs of this data. Among the illicit drugs shown on the bottom half, marijuana was by far the greatest illicit drug used by pregnant women. Next come opioids, and last, cocaine. This data really begs the question, what is going on with the developing fetus in these mothers? Cannabis exposure to the developing fetus has been linked to deleterious effects or negative developmental effects. It increases the chance of abortions or premature birth. And it has also shown to have short and long-term effects. For example, a short-term effect is less than normal weight of the baby at birth. It can decrease the level of dopamine D2 receptors in the brain, which cause long-term effects. This can cause uncontrolled tremors, attention deficit hyperactivity disorder, and Tourette's syndrome. These effects can be seen through the life of the person into adulthood. As you can see, it causes neurodevelopmental disorders, which causes lower performance in school and a reduced short-term memory. Behavioral issues have also been observed, such as aggression in females, depression, impulsivity, and inattention. As the child ages, these behavioral issues can lead to delinquency and mischievous behavior. Finally, in adulthood, it can cause drug-seeking tendencies and a reduction in visual spatial memory. An example of this is not being able to, say, judge distances, say, between your car and the car next to you on the road, which we can all agree that is very dangerous. Cannabis use during pregnancy also leads to legal consequences for the mother. There's actually a law in place called the Keeping Children and Families Safe Act. That requires a physician to notify the Child Protective Services Agency if they suspect drug exposure has taken place. All of these reasons created really the motivation for us to be able to track cannabis exposure of the fetus during pregnancy. Currently, there are no specific federal guidelines surrounding maternal or newborn drug testing in the U.S. An objective protocol can help accurately identify cases of exposure and prevent discrimination in testing, such as subjecting different women to different tests. Currently, common methods of tracking cannabis use during pregnancy are self-reporting of mothers in their regular doctor's visits, and they report it to their doctors, and questionnaires. But the first two, as you can imagine, they're unreliable. They may be quickly collected, they have little cost to them, but because of the fear of legal trouble, much underreporting exists. Drug testing of urine is another method. The collection is also quick, it's non-invasive, it's waste material. Although these practices provide information of drug use, they don't definitively prove that the drug reached the fetus. This may cause wrongful convictions. And there's actually one case recently where a mother's urine was tested in a hospital and it tested positive. But this mother knows she doesn't do drugs. But that didn't stop them from starting legal proceedings on her and hitting her with fines and calling Child Protective Services until she hired a lawyer. And the lawyer was able to initiate an investigation that found that the test was a false positive. And so this really does present the problem. These are reasons for why it is important that we explore other methods of detection. And a good place to explore are specimens that come directly from the pregnancy that have the potential of offering clear evidence of fetal drug exposure. The transfer of biological fluids between the mother and developing fetus is a constant exchange. Here we see some biological materials that can offer us information related to drug use of the mother. The biological specimens we see on the left are known to show the presence of drugs in a drug user. And one of those specimens is blood. In a pregnant woman, blood and other fluids transfer from mother to placenta. That we see on the right here as a pink half circle, then to the umbilical cord, to the fetus, to the amniotic fluid that surrounds the fetus as well. And so it makes sense that if the biological material produced from pregnancy can show level of drugs if used by the mother. Everything on the right half of this picture are all biological specimens that come out at the date of birth. Among them is meconium, umbilical cord, and the placenta, which are the fetal specimens we will focus on, as well as the mother's hair and urine. Testing postpartum specimens with a long window of detection provides a better opportunity for detecting drug exposures. But what is this window of detection? The window of detection is a range of time that it tells us how long the drug exposure took place. You know, each specimen has its own window of detection. For example, with hair grows at an average 1 to 1.5 centimeters per month. Roughly every centimeter of hair can provide us with a month of drug use information. It's actually very interesting. If we drug test, say, a segment of hair maybe 12 centimeters away from the scalp, we can see whether drugs were used by that person a year ago and which kind of drugs. You've heard me mention meconium as well. Meconium is the stool that begins to collect in the fetus's colon during pregnancy. The meconium is excreted by the baby after birth. Past studies have shown that it has a detection window starting at some point in the second trimester. However, it can be unreliable since meconium can become dislodged in the womb, rendering it useless for analysis. The umbilical cord has shown a window of detection beginning at the third trimester. A benefit of the umbilical cord is that it's waste material after birth and it remains intact. Maternal urine also provides drug information, but has a short window of detection of about 30 days for a chronic or very frequent user, and as little as 3 days for an infrequent user. The placenta detection window is currently unknown. I think the placenta is an interesting organ to study for cannabis exposure, since it's the first checkpoint between mother and baby. Here, we can see an actual placenta inside the amniotic sac just after birth. As we can see, it's actually pretty large. But what does the placenta actually do? It's an organ that develops during pregnancy that allows the transfer of oxygen and nutrients from the mother to the fetus. It allows also waste material from the fetus to pass. So it allows a constant exchange of fluids in both directions. This organ actually performs a lot of hard work. On the right here, we see a magnified section of where the umbilical cord is connected to the placenta. If we look at the right side, the mother's blood vessels transfer blood and nutrients to what is called the placental alveoli. Those are the sacs we see that resemble sort of like a tree, leaf from a tree. This sac absorbs the blood and nutrients and transfers them to the fetus's blood vessels, which then flow through the umbilical cord to the fetus. That way, they get all that like yummy Big Macs and Subway sandwiches the mom eats, or at least just the nutrients from them. That would be pretty cool if the fetus can taste the mother's pancakes every morning, huh? In addition to all that, the placenta also performs other necessary functions, such as hormonal and metabolic functions. So similar to what I discussed earlier, where THC is converted to other metabolites by the liver, the placenta is also capable of performing the same function on the nutrients coming from the mother. And as we see in the photo on the left, it is found in abundant quantity. And due to it being waste product after birth, the collection is non-invasive. All of this information really made us optimistic in the studying of placenta. Also, it's important to note that in previous studies, cannabinoids were found in umbilical cord and meconium, which is why it led me to hypothesize that based on prior evidence that shows the presence of cannabinoids in meconium and umbilical cord, cannabinoids will be present in the placenta. And to us, it made a lot of sense, you know, if the placenta is the first checkpoint between mother and fetus, and drugs were found in places that come in line after the placenta, then the placenta would show levels of drugs, right? Makes sense? It had to pass through there at some point. And just how do we go about tackling, you know, finding out whether placenta does show any presence of cannabinoids? I took a research approach that aimed at taking raw placenta and creating an extraction procedure that would result in a final liquid sample that contains the cannabinoids of interest, to then submit that sample for instrumental analysis that's capable of detecting and measuring the levels of the cannabinoids that are present. Once a good method was developed and strict toxicological standards were met, the method was used to test authentic placenta samples from women volunteers who donated and who have reported having used cannabis during pregnancy. To begin tackling the extraction of the cannabinoids, we had to do just that and begin with an extraction technique. The placenta is a solid tissue. Our first goal is to make it into a homogenate. A homogenate of placenta, I guess, is best described as a placenta like smoothie. That may sound funny, but that's exactly what it looks like. I also want to make something clear. In this stage of the project, I am using clean placenta that does not have drugs from the donor. So what we do is, well, we take solid placenta and we cut it into tiny pieces and weigh out 500 milligrams to be used in our extraction procedure. We then place it into a test tube with ceramic beads, as seen in the second picture here. We also mix it with the solution that contains the cannabinoids of interest that we're after, pictured here as the yellow stars. The bead mill homogenizer seen in the center then shakes the tube vigorously so that the beads can mash, grind, and blend the placenta. The result is a sample with uniform distribution of the contents of the sample. This is the placenta smoothie at this point. It has the color of a strawberry shake, but we're not done yet here on at this point. We have to purify the sample by getting rid of the solid mush that's in the tube. So we use a centrifuge. Pardon me. We use a centrifuge that we see in the fourth picture here. And this centrifuge spins the sample at a very high rate of speed. This results in a separation of the liquid layer on top and a solid layer at the bottom. So now the liquid layer contains the cannabinoids we want, and also some components of the placenta such as fats, proteins, and other interferences that we don't want. And the liquid portion is collected, as seen in the last image, for a solid phase extraction technique, which further purifies the sample. And the goal of solid phase extraction is to further purify our sample. It starts with a sample that has our drugs and interferences, and it ends with a sample with only our drugs. So to do this, we use a cartridge with a sorbent bed. And the sorbent bed is shown here as the brown square portion of the cartridge. This sorbent bed is made up of chemical compounds that attract the cannabinoids and make them stick to the bed. This allows for the washing away of interferences that we don't want, so that we can collect later the cannabinoids we do want. So we begin by prepping the cartridge as seen all the way to the left-hand side. And we prep it first by passing a solvent and water through it. We do this to the cartridge for the same reasons you want to add, say, dish soap to a wet sponge and not a dry sponge before you begin washing your dishes. Otherwise, you lose a lot of soap on the first plate as you start scrubbing. Here, we would lose a lot of our drug if we don't prep the cartridge. So we prep the cartridge, then we add our sample and allow it to flow through. We can see in the second step how the cannabinoids depicted as the yellow stars stay stuck to the sorbent bed. At the bottom of the cartridge, we also have interferences in red stuck to the bed. And to get rid of these interferences, we follow up with washing steps where we use solvents designed to only pull out the interferences while leaving behind the cannabinoids. That can be seen in the third and fourth step. The first wash, depicted as the little black boxes, is able to remove some interferences. The next wash, shown as the red triangles, pulls out the remainder of the interferences. Finally, a solvent is used that is strongly attracted to the cannabinoids to pull out the cannabinoids, and that's seen as the little pink circles. This is the sample that we collect. What we actually don't want this particular solvent we just used in our sample for later when we test in the instrument. What we have to do is evaporate the solvent, and this is done in a machine called a turbo evaporator, or for short, turbovat. The turbovat has a hot water bath where the tube sits while nitrogen gas is blown into the tube to speed up the evaporation. This helps cut back on a lot of time in the analysis. Once the turbovat step is complete, the dry sample is reconstituted or simply dissolved in the solvent used for the instrumental analysis. The first liquid seen in the blue cap vial all the way on the right-hand side is the usable liquid sample we've been after this whole time. This is the sample we use to test in our analytical instrument. Here's our analytical instrument, a liquid chromatograph tandem mass spectrometer. The goal of this instrument is to separate the mixtures to each of their individual components, followed by the identification of each component and the measurement of each component. In the field, we call this a hyphenated system because it brings two instruments together to form one. This improves resolution in the separation and in identification. Right now, let's focus on the liquid chromatograph portion of the instrument, which is the two-tower system on the left-hand side. The sample we yielded from the solid phase extraction in the little vial is injected into the liquid chromatograph and is carried through a system of pumps and hoses to reach the analytical column seen here as the long cylinder. The yellow stars on the left represent the cannabinoids all mixed together as they naturally are in our sample. But as they travel through the column, they separate as seen by the different color stars. This is possible because the analytical column contains inner walls with chemical compounds fused to those walls that will attract the cannabinoids as they travel through. Each cannabinoid is attracted to the wall to different degrees. The most attracted will be the last to exit, and the least attracted would be the first to exit. When they exit, they enter the tandem mass spectrometer. Now our sample has been separated, and the cannabinoids are leaving the liquid chromatograph one by one and entering the tandem mass spectrometer that we see above here as the long box. The tandem mass spectrometer is capable of identifying the cannabinoids based on their masses with the use of two mass analyzers. Similar to those tandem bicycles that we see on the beach where there are two riders, here we have two mass analyzers in tandem, hence the term tandem mass spectrometer. The first cannabinoid, as seen as the red star on the left, enters the electrospray ionization chamber. Here the cannabinoid, which is still a liquid sample, is converted to a gas given an electric charge, making it an ion, and is sent through an electric field. This ion, we call the parent ion. The first component in the electric field is the first mass analyzer, and it's seen as the 4 rods. It is set to operate at an alternating current that keeps the parent ion suspended in space so that it can travel through. And it travels through in sort of a swirl motion, seen as the swirly red line there. Or for you baseball fans, it flies through like a knuckleball. And if you aren't familiar with baseball, folks, you can all Google that later. The term is knuckleball. So it goes through the first mass analyzer and makes it to the cylinder in the center called a collision cell, where the parent ion collides with an inert gas, causing it to fragment. These fragments we call daughter ions. The daughter ions then go through the second mass analyzer and reach the detector. The detector then transmits the signal to the computer software, and the computer software produces a graphical picture like the one seen on the right. The transformation from parent ion to daughter ion is called transition. This transition is how the instrument is able to identify a compound, because each compound fragments in its own special way, providing a fingerprint of its identity. As more of the cannabinoids are sent through after they come out of the liquid chromatograph, we get a collection of graphical data. This here is an example of the collection of graphical data generated by one of our samples at a concentration of 30 nanograms of cannabinoids to 1 gram of placenta. Each graph shows the signal from the mass spectrometer when the chemical compound began collecting at the detector. The more cannabinoid detected by the instrument, the more pronounced of a peak is formed. And as we can see, each cannabinoid in this particular sample was successfully detected. With the analytical method finally developed, we applied the method to authentic samples. This project is done in collaboration with the University of Santiago de Compostela in Spain, and is part of a larger study which investigates alternative matrices to detect in utero drug exposure. Our authentic samples were collected between the years 2011 and 2015. In the larger study, about 800 volunteers donated samples. Out of those, 25 maternal hair samples tested positive for cannabinoids, as well as 13 meconium and 12 umbilical cord. But what about placenta? Well, that was left to me to find out, and it was my job to find out. Before I explain these results, I like to just say that we were only able to test 7 cases, and this is due to the coronavirus shutting down of all CUNY campuses, and it threw a wrench in our project. But here we are. Each case listed on the left represents 1 volunteer, and each of them donated the specimens that are listed in the columns to the right. As shown, all cases tested positive for cannabinoids in the umbilical cord and meconium, and 5 maternal hair specimens tested positive, while 2 tested negative. In my project, I discovered cannabinoids in all 7 of the placenta samples. I find it fascinating that the cannabinoids actually remain preserved in the placenta for about 8 years since their collection. As you can see, they were collected in 2012. Another interesting thing is the negative result for the hair, but the positive for the fetal specimens of the same volunteer. This really takes us back to earlier when I discussed the windows of detection. Could this be that the mother began using cannabis just outside that window of detection of the hair? That would be sometime late in the 3rd trimester or close to the date of birth. I think that would be something very interesting to follow up with the contributors from Spain. And what these results actually told us is that the method worked. And so with the agreement of our placenta results to the other specimens in our 7 cases, we successfully developed a specific and sensitive method for the determination of THC and metabolites in placenta by liquid chromatography tandem mass spectrometry. And the method was validated based on current toxicological standards as well. For more on the validation process, you can tune in later in the toxicology presentation room, as my co-contributor Frank Gutierrez will describe it in more detail. The positive placenta results showed the usefulness of using placenta to detect cannabis in utero exposure. And finally, my hypothesis that cannabinoids will be present in placenta because it was found in meconium is accepted. And I would like to thank the contributors from Spain for making this project possible, the PRISM program and John Jay for their funding, and my friends that worked side by side with me in the John Jay Toxicology Lab. They're all pictured here. None of them are left out. You guys may not know this, but you all provided some emotional support for me along the way throughout the year and a half, and much technical support as well. Thank you. Thank you so much for that. Most of us are actually going our separate ways after this month's graduation, and I will miss you all dearly. Thank you very much. That concludes the presentation.
Thank you very much, Alejandro. Good evening, everyone. I am Dr. Edgardo Sanabria Valentin, and I am the Associate Director of PRISM. I will be conducting the Q&A session, for which we have around 10 minutes. Alejandro, every year I reserve the right of asking the first question, so here it goes. In our audience, we have members of next year's new batch of PRISM undergraduate researchers. What have you learned by participating in this project that you would like to share with others, with them?
Wow, there's so much. Well, first of all, congratulations on surviving an interview with Dr. Ed and Dr. Sanguinetti. It could have been very intimidating, as it was for me. I actually paced back and forth outside of their office before they called me in to relieve some of the nerves. Well, you really, that pacing back and forth won't end. In your research, you won't always get the results you hope for, you know, and you always have to tap back into your memory of everything you've learned in all the courses before you at that point. And well, just take it back here on what scientific research really is, right? You're taking something from nature and you're putting it into a little environment that you created, say maybe in a test tube, a petri dish, or a plexiglass cage so that your I don't know, insects won't fly away. And you are observing what is happening to this thing that you are studying and to the environment that you created. And those results you have to observe and really interpret. And you can't make up any results. So, like I said, they won't always be what you hoped for. So you always have to read more literature, see maybe where you may have gone wrong, and revamp that experiment. But it's all fun. As you can see, mine took about a year to do, and there was a lot of that. We didn't get the results we wanted. We had to go back and study just how these cannabinoids may interact with certain things in our process. Then we had to change some of that process. And after months, I'd say maybe 7 or 8 months, we finally hit it on the head and ended our project. But it's all fun. It's stressful, but it's definitely rewarding at the end.
Okay. I have the following question from the audience. If the placenta is considered to be biomedical waste, does the mother have to give permission to the scientist to collect it?
I don't want to pass myself as as any expert in there. I don't know. I don't know, Dr. Concheiro, maybe
yeah,
you would know that.
I can help you out. It's my pleasure. No, definitely. Every time you are getting a biological sample from anybody, you need the consent. And for example, in the study that we conducted with the university in Santiago, all the mothers gave their consent orally and written, and they were informed about everything that we were going to do, how long we were going to keep the samples, every test we were going to do. So definitely everybody has to be informed 100%.
Okay, I have the following question. Would you be able to tell, based on the tox analysis, whether a mother was a chronic user versus an infrequent user of cannabis?
Well, what I really studied in the literature was just if she was a user, whether it was frequent or not. No, I'm sorry, I don't know the answer to that.
Yeah, I don't know if you want me to add a small comment. Alejandro is totally right. We cannot say if it is chronic or sporadic. That's why having all these different matrices with the hair, with the meconium, the core, the placenta, help us to figure out how often that consumption was. But just for testing the placenta is impossible, unfortunately.
Okay, I think we have time for a couple more. I have a question from a member of our faculty, actually. So if you could design a future experiment with all things possible, within ethical limits, of course, so that you could test your hypothesis, what would it be? You have every resource, you had $1 million grant from the NIH to do this.
Oh, man. Oh. Well, I hate to dip into that last question, but that really sounds really interesting to me, to test whether the person was a frequent or infrequent user. And in addition to that, also, what is the case with, say, a person that only experienced secondhand smoke? So if a pregnant mother may have been living in an apartment or a home with others that used cannabis, how could that have entered her, and could that have gone into the fetal specimens there?
Okay, then last one. I have a question here. Were the placentas used for the experiment from cannabis users only, or do you know if they were using other drugs?
These they reported cannabis use and also tobacco. But in the larger study, there were also specimens of people that used opioids and cocaine. And those produced similar results to my project, where they also tested positive in the placenta for those drugs.
Good. I have one last question. Someone wants to know, what are your future plans after you graduate from John Jay?
Well, in the current state of our planet as it is, I can't go on vacation. So I'm going to move closer to where I was accepted to graduate school in Stony Brook on Long Island. And I'm actually interviewing for a few lab jobs, some lab tech. jobs around the area, so it could make my moving a little easier. And I guess, you know, working a full-time job while going to school is still going to be my reality. That's where I see myself in the next few months.
Thank you, Alejandro. That was great. That concludes our first plenary session, but don't leave yet, because we're moving to the second part of our e-Symposium. Our undergraduate researchers were presented with a challenge 4 weeks ago. Prepare a 5-minute presentation about your research using 10 slides or less, and tell the story of your research to a general audience. We were inspired by the 3-minute thesis competition, and I can tell you, our students have done a great job.